today is Dr. Jeff Iliff. He is a professor of psychiatry and neurology at the University of Washington School of Medicine. His earlier work helped to define the glymphatic system in rodents, and more recently, his group has been an important contributor extending this biology into the human brain and understanding its role in the development of different neurological and psychiatric conditions. Welcome, Jeff.
Great, thank you. Looking forward to the chance to speak to this group. Can you see my screen okay?
Yes.
Great. So over the next 40, 45 minutes or so, what I'll be doing is walking through how we've come to understand the glymphatic system as a sleep active biology generally in rodents and then in humans, and then kind of taking us a bit of a status update on where we're at in terms of understanding its role in the pathogenesis of Alzheimer's disease, focusing on distinguishing between things that we think and things that we know, which is sometimes a distinction that isn't always appreciated. A couple disclosures. One that's relevant is I do serve as the chair for the scientific advisory board for a company called Applied Cognition that are active in the glymphatic space. They pay me, and I own a small portion of the company, and I will be presenting data that was developed in collaboration with that group. So you need to know about that conflict. I am a full-time U.S. federal employee, which is pretty exciting these days. My opinions are not that of the U.S. government, and the U.S. governments are not mine. So there we are. So I find when I talk with an audience, and I use the word glymphatic, the word glymphatic very often. If I have 10 people in the audience, there are 10 different ideas of what the heck that means. And so what I will do first is sort of level set. When I use the word glymphatic, this is what I mean, so that you have understand my lens as I'm approaching this biology, just because I find that that word is sometimes a little bit of a Rorschach test in terms of what people think about the brain and fluid dynamics and waste clearance and things like that. So how I would define glymphatic function and glymphatic exchange is encapsulated on this slide. So I would say that over the last 13 or almost 14 years now, data from rodents, mostly, increasingly human work though, has demonstrated that the fluid that surrounds the brain, the cerebrospinal fluid, it doesn't just sit there passively. It isn't simply a cushion or a medium in which the brain floats, but rather it exchanges dynamically with the brain's interstitial compartment, so the fluid between the brain cells. That movement of fluid into the brain and the clearance of fluid and solutes out of the brain appears to be anatomically organized, meaning following along the outsides of blood vessels through what are termed perivascular spaces, and rapid. It's more rapid than is accounted for by diffusion alone, so it appears to have a substantial convective or bulk flow component to it. It appears to be driven by different oscillatory cycles, including arterial pulsation, maybe respiration, certainly low frequency vasomotor oscillation, so intrinsic dilations of blood vessels, and also synchronous neural activity that is characteristic of different sleep states. It appears to be supported by the astroglial network and astroglial water transport in particularly. So the astrocytes express a water channel called aquaporin 4 that is localized primarily to perivascular astrocytic end feet, and that perivascular aquaporin 4 seems to play an important role in supporting this perivascular transport. Data starting in 2013 and extending through present day suggests that this is a biology that is active primarily in the sleeping compared to the waking brain, although that's a little bit controversial these days. This glymphatic exchange, so that's the exchange between the CSF outside the brain and the interstitial fluid within the brain, appears to be closely related to the uptake of solutes and fluid from the CSF into meningeal lymphatic vessels. So in 2015, lymphatic vessels were characterized in the meninges surrounding the brain. They take up solutes from the CSF. And they take up CSF also. We believe that the function of glymphatic exchange and meningeal lymphatic drainage are coupled to one another and likely co-evolved with one another. And data from rodents and some data from humans suggests that this clearance contributes to the clearance of amyloid, tau, and synuclein, which of course are the proteins and peptides that aggregate in the setting of different neurodegenerative conditions. So that's the glymphatic system as I understand it. Of course, I just told you a whole bunch of things. I showed you zero data supporting those things. As many of you are scientists, I suspect that you know that no one can lie about data quite so much as a scientist discussing their own data. So you shouldn't take my word for those things. If you want to go check out the articles that support those statements that I'm making, if you take out your phone and you go to this QR code, it will take you to a list of articles in PubMed that support these statements that I'm making. Among these statements, two of them in particular, this idea that clearance is more rapid in the sleeping brain and contributes to the clearance of wastes like amyloid, tau, and synuclein, that has led to the widespread belief that, and here I say belief, and not I wouldn't say that this has been proven, that glymphatic clearance, at least in part, supports some of the restorative effects that sleep appears to have on cognition. And in that impairment of this process of the sleep active glymphatic clearance appears to, or is believed at least, to underlie the observed clinical association between sleep disruption and dementing disorders such as Alzheimer's disease. These are beliefs, and what I want to do over the next little while is walk through whether this is true or not, or how much data do we have to actually support these claims, and how solid are these claims. So first, what is the preclinical evidence that glymphatic impairment contributes to the development of Alzheimer's disease? So I am a person, I have a pretty substantial preclinical rodent program, but I am also a person who's fairly skeptical of rodent models of neurological conditions, whether that's traumatic brain injury or stroke or Alzheimer's disease or Parkinson's disease. And that's driven partly by my experience. I did my PhD in a stroke lab back in the early 2000s, that many of the things that appear to improve outcomes in rodent models of neurological conditions don't end up translating. So I think that there are some questions that animal models can answer really well, and there's a lot of questions that they can't answer well. So as I think about what we can learn from preclinical models in the setting of AD, first I think about this is I go to the lens that I go to the lens that I approach these conditions with. So first I think about Alzheimer's disease, particularly neurodegenerative diseases generally, as proteinopathies. They are diseases of protein misaggregation. So in the case of Alzheimer's disease, this is a condition that is characterized histopathologically by the presence of extracellular plaques made up of amyloid beta, intracellular tangles made up of hyperphosphorylated versions of the protein tau. So these are these are diseases of protein misaggregation. But these are also conditions that are influenced by different non-genetic risk factors. So in their sporadic form, diseases like Alzheimer's and Parkinson's are influenced by different non-genetic risk factors. This includes aging, which is the primary risk factor. In fact, it's such a big risk factor that you don't even think about it as a risk factor. Other chronic medical comorbidities like hypertension and diabetes, comorbid cerebrovascular disease, traumatic brain injury, chronic sleep disruption, all of these appear to be risk factors for the development of Alzheimer's disease. And the thinking is that there is something about the aging brain, something about the hypertensive, the injured, or the sleepless brain that makes it vulnerable to this process of protein misaggregation. And that's why these are risk factors. So I think as we think about these conditions, I think there's three general questions that rodent models can help us answer as we consider whether glymphatic impairment contributes to the development of Alzheimer's disease. So the first is, do we actually see evidence that the glymphatic system contributes to the clearance of amyloid and tau? Is it involved in the process of amyloid and tau clearance? We can address that question in animals. We can ask the question, is glymphatic function impaired in these non-genetic, in the setting of these non-genetic AD risk factors, right? So if we see that glymphatic function is impaired in aging or hypertension or diabetes in animals, then that this is consistent or supports the idea that glymphatic impairment is the mechanism that may link these risk factors to vulnerability to amyloid and tau pathology. Lastly, and maybe more mechanistically, we can answer the question of whether experimental manipulation, like experimental impairment of glymphatic function, is it sufficient to promote the development of amyloid or tau pathology, right? So if we modulate glymphatic function, can we drive pathological burden? If we can, then that supports the idea that that impairment of this process contributes to these diseases. So we've actually made a fair amount of progress in answering all three of these questions. This is exclusively at this point in published data, so I'm only going to treat each of these really briefly in sort of a vignette form, and then I'll take you to a QR code later on that'll allow you to go check out these things in more detail. So does the glymphatic system contribute to the clearance of amyloid and tau in rodents at least? And the answer to that seems to be yes. This is an example of a study looking at that question. This was our initial characterization of glymphatic function back in 2012. In this study, one of the things that we did is we injected fluorescently labeled amyloid into brain tissue, and we saw that it moved through brain tissue and is cleared from brain tissue along perivascular compartments. So there is perivascular transport of amyloid. We observed we have a transgenic animal model, which is the aquaporin 4 null mouse. So this is a mouse that where the gene encoding aquaporin 4 is disrupted. We observed that this animal has impaired glymphatic function, right? So this is the initial data suggesting or demonstrating that glymphatic function is dependent on aquaporin 4. We see that if we inject in radiolabeled amyloid beta 40 in wild-type animals, maybe 70% of that amyloid is cleared in the first hour. And we observe that in the setting of aquaporin 4 gene deletion, that clearance is slowed. So these data support the idea that amyloid beta is cleared by the glymphatic system. Other data, and both from our group and from other groups, has demonstrated a similar role for glymphatic function in the clearance of tau and the clearance of alpha-synuclein. So check. We think that it does appear that the glymphatic system contributes to amyloid, the clearance of amyloid and tau. Is glymphatic function impaired in animal models of non-genetic AD risk factors? The answer to that also seems to be yes. So this was a study that this is an example study, something we published back in 2014. So more than 10 years ago now. This is simply asking the question, is glymphatic function impaired in the setting of aging? No other comorbidity, so no cerebrovascular disease, no traumatic brain injury, nothing. These are just older mice. So if we look at the movement of CSF tracer into brain tissue in healthy young adult mice, 2 to 3 months of age, and older mice, 18 months of age, we see that the movement of CSF tracer into the aged animals is markedly reduced. If we measure the clearance of amyloid out of brain tissue, we observe a similar slowing with increasing age. So in healthy young animals, in the first hour, maybe 70% of the amyloid is cleared. In 12-month-old and in 18-month-old animals, we see a stepwise slowing in the clearance of amyloid. So in at least aging animals, we see there's impairment of glymphatic function. And I will tell you that in other studies that our group and a number of other groups now have reported, we do observe impairment of glymphatic function in animal models of hypertension, in animal models of diabetes, animal models of cerebrovascular disease, animal models of traumatic brain injury, including mild traumatic brain injury, animal models of chronic and acute sleep disruption. So is glymphatic impairment observed in the setting of non-genetic AD risk factors in animal models? The answer seems to be yes. So the next question, does experimental impairment of glymphatic function promote the development of amyloid or tau pathology? This is one study that we did. A number of groups have addressed this question. This is simply one example. So in this, what we did is we took the 5XFAD mouse line. So this is a transgenic overexpression line that produces amyloid plaques very early in life. We crossed it with the aquaporin 4 null line. We did a heterozygote cross so that we would end up with 5XFAD mice with aquaporin 4 and 5XFAD mice without aquaporin 4. Measure amyloid plaque burden histologically. And what we observe is that in the animals with aquaporin 4 intact, we see a certain level of pathological burden, so of amyloid plaque burden. And we see that if we knock out aquaporin 4, we see exacerbation of this pathology. This and a number of other studies demonstrate that if we impair glymphatic function experimentally, it is in fact sufficient to drive the development of amyloid, tau, and other studies actually show the same is true of synuclein pathology. So check. Again, I showed you basically 3 pieces of data here in these last 3 slides. That represents really dozens of studies that have demonstrated these things across a number of different preclinical, mostly mice, but some rat studies that I think are worth going to look at. So if you want to check that work out, you can follow this QR code. It will take you to a new list of papers which support these 3 claims, of which the ones that I just showed you are examples. So everything I've shown you so far is data from rodents. As I mentioned before, I am frankly a little bit skeptical of rodent work, even though I do rodent work. I think a lot of things that we find in rodents don't turn out to be true in people. And so the question of whether glymphatic clearance is a feature of the human brain and whether impairment of that clearance contributes to the development of clinical Alzheimer's disease, I think that is an important and separate question. So the first thing I'm curious about, or that we wanted to try to figure out, is sleep-active glymphatic clearance a feature of the human brain, or is this just something that we see in rodents? So glymphatic function has mainly been up until 2015 and up until actually very recently, glymphatic function has mostly been characterized in rodent models. The role of sleep and in regulating glymphatic function and the features of sleep that regulate glymphatic function has really been has mostly been characterized in these 4 papers. So a couple papers in 2012 and 2013, a slightly more recent paper in 2019. So what we're going to do is do a little bit of a mini journal club, walking through the findings that characterize sleep-active lymphatic function in rodents from these 4 papers. And then we're going to look at the studies that have shown parallel findings in the human brain, right? So where are we at in terms of understanding the sleep-active lymphatic function in the human brain compared to what we know in rodents? So the first thing that we learned about glymphatic function in mice came as a result of the use of dynamic contrast or tracer-based imaging, right, in living animals. So that was dynamic 2-photon microscopy in vivo in mice. It was dynamic contrast enhanced MRI in mice and in rats. These 2 imaging modalities allowed us to see 2 important things about glymphatic function. So one, it was that the movement of fluid and solutes into and through the brain is anatomically organized around the scaffold of the cerebral vasculature. So this is a study that we did in healthy young mice using 2-photon microscopy through a cranial window. We labeled the vasculature with a red tracer. We then injected in different tracers into the CSF. And what we observed when we looked at the brain surface was that the movement of tracer initially moved along the brain surface along pial arterioles. If you look up close, it's through some sort of CSF subcompartment. So through these perivascular spaces of the brain surface. And then as these blood vessels turn and dive through the cortical mantle, we see that the CSF tracer follows through these perivascular spaces surrounding the blood vessels. And then we see that those tracers eventually exchange through the entire brain, through the entire cortical interstitium. So this demonstrated that perivascular transport or organization around the cerebral vasculature was a major organizing principle for fluid movement through the brain. Contrast-enhanced MRI being intrinsically three-dimensional, gave us the ability to evaluate whether this is something that's happening in just some brain regions, or is this happening across the entire brain volume? And what we observed is with contrast-enhanced MRI is that this CSF contrast exchange was happening really throughout the brain. So this is a brain-wide exchange. The interesting thing is that contrast-enhanced MRI, that's an approach that we can use in rodents, but it turns out it's also an approach that can be used in humans. I'm sorry, yeah, we can do it in rodents, but it can also be done in humans. So we can inject gadolinium-based contrast agents intrathecally, so into the CSF that surrounds the CNS. This is an off-label use of a contrast agent, right? So it's not FDA approved, but it can be done when clinically indicated and at a physician's discretion. So there's a group in Oslo in Norway, so Per Christian Eide and Geir Ringstad. They've since 2015, they've been doing contrast-enhanced MRI in people to measure glymphatic function. And using that technique, they have been able to validate a number of the key physiological features of glymphatic function defined in rodents in humans. So these are two examples of work that they've done. So this is actually a fairly recent piece that they published in Nature Communications, where they used an approach with high temporal resolution and high spatial resolution to demonstrate that the movement of contrast from the cisternal CSF compartments, initial movement of contrast into brain tissue, it does indeed follow these perivascular pathways. You can see the dark haloed pathways. These are cerebral arteries entering into brain tissue. So this work demonstrated that the initial movement of contrast, CSF contrast into brain tissue in humans does in fact follow along these perivascular pathways following the cerebral arterial vasculature. Earlier studies that they had done had shown that this is a brain-wide process. So this is a case series that they published in Brain back in 2017, where they followed contrast enhancement over a number of hours. And so what they observed is prior to contrast injection, so this is the MRI scan pre-contrast, 1 hour, 3, 4.5, 24 hours after contrast injection. What you can see is early filling of the cisternal CSF compartments with the contrast agent. You see early enhancement of brain tissue. These data suggest that early enhancement is perivascular initially. And then when participants came back the next day, what you observe is global enhancement with contrast. So what this demonstrates is that this exchange of fluid through fluid and solutes is occurring really through the entirety of the brain volume. So this is a brain-wide perivascular exchange. Just like what we saw in rodents, this group in Oslo has confirmed that we see the same thing in humans. Another observation, another key finding that was made along the way was that this process of exchange, of CSF interstitial fluid exchange, is sleep active. So that was initially described in this paper published in 2019 in Science. The observation is pretty straightforward. If you take an animal that's asleep, so this is actually recovery sleep following sleep deprivation, and you measure CSF tracer movement into brain tissue, we see that in the sleeping condition, there's pretty rapid movement of tracer movement in. If you awaken the animal and inject a second tracer, you see the movement of that second tracer into that same animal is slowed, suggesting that the movement of tracer into brain tissue is sensitive to sleep deprivation, so it's sleep dependent. They see the same thing with the clearance of solutes out of brain tissue. So in this case, you're injecting a solute, in this case, radiolabeled inulin, into brain tissue, and then you're measuring its clearance out over time. And what you observe is that the clearance of solute out of the sleeping and the anesthetized brain has a certain speed or a certain kinetics to it. And that if you awaken the animals, you can see that the kinetics of clearance out of the brain tissue is markedly slowed. So both the movement of CSF into brain tissue and the clearance of interstitial solutes out of brain tissue is sensitive to sleep deprivation. That's a finding that stood in the field for 8 years and prior to its eventual validation. So this is now 2021. This is that group in Oslo again, using intrathecal gadolinium contrast enhanced MRI. And so this is the study design here is actually pretty straightforward. So they took a number of participants. They randomized them into 2 groups, in this case, the red and the blue group. They carried out MRIs in the late afternoon, early evening to measure how much contrast had loaded into brain tissue. And what they observed was that loading between the 2 groups was roughly equal. So there's an equivalent amount of contrast in the brain tissue of these people. And then what happened was the red group was sleep deprived overnight, and the blue group was allowed to sleep normally in the hospital overnight. And then when the participants came back the next day, they scanned how much of the contrast that had loaded into the brain tissue at this time point had been cleared, right? So in the participants that slept normally overnight, you saw a certain rate of clearance out of the brain. And you saw that in the participants who were sleep deprived overnight, you saw an attenuation of that clearance. So this was the first validation, not just in another lab, but in another species, that the clearance of solutes out of brain tissue is indeed sensitive to sleep deprivation. Even when both of these groups were allowed to sleep in the second night following, that deficit in clearance that resulted from that first night, it still persisted, right? So it took more than a night of recovery sleep for this deficit to be erased. So those data suggest that sleep active glymphatic function is in fact a feature of the sleeping human brain. The question is, what is it about sleep? Which sleep-related physiological features are driving this increased function? So 2 studies have given us some important physiological determinants of this sleep active glymphatic function in rodents. So the first I'm going to highlight from this paper that was published in 2019. This is really kind of a heroic experiment that Lauren Hablitz carried out when she was in Micah Niedergaard's lab in Rochester. So she injected a whole bunch of animals with fluorescent tracers and measured glymphatic function into brain tissue. The difference being that each of the animals were anesthetized with different anesthetics. And so what she observed, and this is something that many of us had noted anecdotally in our own work, but then she sort of systematically went through, was that some anesthetic regimes resulted in enhanced glymphatic function. So ketamine, xylazine cocktail, dexmedetomidine on the backdrop of light isoflurane anesthesia, those seem to enhance glymphatic function. While other anesthetic regimes like Avertin or isoflurane seem to suppress it. What this told us was, well, the anesthesia that you use when you're studying glymphatic function matters a lot. What she also did, though, in these same animals was measure the EEG spectra. So she measured the electrophysiological state of the animals while they're under these different anesthetic states. And what that allowed her to do is to look at, without regard to anesthesia, what are the EEG spectral features that are associated with enhanced or suppressed glymphatic function? So if she looked at high frequency neural activity, so beta band power, or low frequency delta band power, and compared them and related them to glymphatic function here on the y-axis, so higher function going up, what she observed was that higher glymphatic function was associated with reduced beta power and increased delta power, right? So higher low frequency activity and lower high frequency activity were each associated with enhanced glymphatic function. She also had cardiovascular data, so she observed that the anesthetics that were associated with reduced heart rate, which we think is related to reduced sympathetic and reduced central noradrenergic drive, were also associated with increased glymphatic function. So reduced EEG beta power, increased EEG delta power, reduced heart rate, all associated with increased glymphatic function. Another sleep-related physiological finding that was associated with this sleep active function were changes in the brain's extracellular space itself. So this is a kind of, I think, underappreciated finding from that paper in 2013. So this experiment used an electrophysiological technique that probes the brain's extracellular space. So what it does is it actually measures the diffusion of a cation from one electrode to another electrode. And from the behavior, from the transport of that ion between these two probes, you can infer information about the space in brain's extracellular space in between. It allows you to measure how big that space is. That's this alpha term. So that's the extracellular volume. And it can tell you how complex or how tortuous that space is. That's this lambda term. And what was observed was that under conditions of both sleep and anesthesia, the brain's extracellular space actually got bigger. So as the brain's extracellular space gets bigger, the resistance of the brain tissue itself to fluid flow goes down, right? And that seems to be one of the things that's happening during sleep that's facilitating this glymphatic function. So these sleep-associated features, reduced EEG beta power, increased delta power, reduced heart rate, and reduced parenchymal resistance to fluid flow. The question is, are these things associated with glymphatic function in humans? That's a question that we were actually able to answer in a study that we published just this last summer. So this is data from a paper we published it in Nature Biomedical Engineering this last summer. This was a collaborative study that involves Swati Rane-Lewandowski. She's an MR physicist. Now she's at Kansas. She used to be here in Seattle with us, but she moved, sadly. Miranda Lim, who's a sleep physician down at Oregon Health Science University in Portland. Paul Dagum, who's the founder and CEO of a company called Applied Cognition that developed a device that measures this thing here called impedance spectroscopy, which I'll tell you about here in a moment. So in this study, we used two different measures that this audience might not be super familiar with. One is, I've told you about the use of contrast-enhanced MRI for measuring glymphatic function using intrathecal gadolinium. That's an off-label use. People in the US aren't too excited about doing that. And so that's a thing that, at least with our group, we're not really pursuing. But what we can do is we can inject intravenous gadolinium. And what we know, or what neuroradiologists know from just their own observations and some things that we've studied now in substantial detail, is that when you inject contrast into the blood, a small percentage of that contrast leaks out into the CSF. And of that contrast, a small percentage exchanges with the brain interstitium. So we can take advantage of that CSF leakage into the CSF and that transport into the brain tissue to measure glymphatic function using intravenous gadolinium administration. So we're using IV contrast-enhanced MRI to measure CSF interstitial fluid exchange. A second measure that we're using that you're probably not familiar with is dynamic impedance spectroscopy. So we use this approach to measure the brain's extracellular space, or its resistance to fluid flow. The idea here is, so this uses a wearable device that has in-ear electrodes, so 2 electrodes, 1 in each ear, that's passing a very small amount of current between those electrodes. Now, at very high frequencies, that current moves through brain tissue without regard to sort of through cells. So at high frequencies, current is able to sort of move through cell membranes, essentially as though they're invisible. So it just sort of moves through brain tissue. But at lower frequencies, cell membranes begin to act as capacitors, and that current has to traverse the brain's extracellular space. And so the movement of current through brain tissue at low frequency begins to become sensitive to the brain's extracellular volume. And so what we can do by sweeping current from high frequency to low frequency, we can measure the dispersion as you move across those frequency bands as a way to capture the brain's extracellular resistance to extracellular fluid flow. So this is a way for us to measure resistance to fluid flow in brain tissue. So we made a clinical study design. It's a little bit complicated. It turns out that doing these kinds of studies in people is pretty tricky. So in this part of the study, we used 20 participants aged 50 to 65 years of age, so older individuals, has a crossover study design where participants either came in on one visit for a normal night's sleep And the next night, night they came in for overnight sleep deprivation. The following morning, participants underwent contrast-enhanced MRI. So we did that contrast-enhanced MRI during this period. And during that contrast-enhanced MRI, participants were given an opportunity to be awake if they had slept the night before, or they were given an opportunity to sleep if they had been awake the night before. This split sleep design allows us to look at the effects that overnight sleep has on the influx of contrast into brain tissue. And it allows us to evaluate the effects of this morning sleep or wake opportunity on the clearance of contrast out of brain tissue. So it gives us two different views of contrast movement into and out of brain tissue. These are data from the study, so from those 20 individuals. This is the EEG data, so represented sort of visually. We see that participants in the waking group were awake. Participants in the sleeping group slept, including REM and N3, that's green. We see that the next morning, participants in the recovery sleep group did undergo sleep, and participants who were waking the next morning were awake. Here's where we're measuring the brain's resistance to fluid flow. This is new data, novel data that had never been measured in people before. What we observed is that in the sleeping participants, the brain's resistance does indeed decline with sleep, whereas in the sleep-deprived individuals, the resistance remained high. The next morning, when these participants were sleeping, we see that the resistance begins to decline. And while these participants are waking, we see that the resistance begins to increase. So this validates the data from the original 2013 paper that the brain's resistance to fluid flow is actually changing during sleep. We can then use these data, these overnight and morning sleep periods, to evaluate whether glymphatic function is associated with differences in beta power, delta power, heart rate, or parenchymal resistance to fluid flow. So we can evaluate the things that we know to be true in rodents. We can evaluate those things in humans directly. So looking at the influx phase and looking at the clearance phase, the sleep associated with each of these phases, so the prior sleep for the influx phase and the concurrent sleep during this clearance phase, we observe that indeed increasing delta power does seem to be associated with greater glymphatic function. Decreasing beta power does seem to be associated with increased glymphatic function. Decreasing heart rate does seem to be associated with glymphatic function, and declining parenchymal resistance seems to be associated with glymphatic function. Note that the effects are reversed. So the signs here, positive to negative, negative to positive, positive to negative, right? In these statistical models, that's because in the first case, we're measuring influx, so an increasing signal. In the latter case, we're measuring efflux, so a decreasing signal. So these data for the first time tell us that glymphatic function, and specifically these physiological features of sleep, are the determinants of sleep-active glymphatic function in humans. So in people, glymphatic function does seem to be occurring. It does seem to be organized along the axis of the cerebral arterial vasculature. Other data that I didn't show you suggests that it is, in fact, more rapid than is accounted for diffusive transport alone. It's sensitive to sleep deprivation. And during sleep in people, it's associated with increased EEG delta power, decreased beta power, decreased heart rate, and reduced parenchymal resistance to fluid flow. So if it's a thing that's happening in people, what is the evidence that we have that its failure is contributing to the development of clinical Alzheimer's disease, right? So what's the clinical evidence? So we'll go back to this kind of lens that our group uses for thinking about Alzheimer's disease. It's a proteinopathy that's associated with these non-genetic risk factors. In humans, we think that we can address maybe 5 different questions as we think about glymphatic dysfunction's role in Alzheimer's disease. The first is in neuropathological studies, so in brain tissue, do we see that there are changes in the human Alzheimer's brain that in mice are related to glymphatic dysfunction? Do we see that? So in a mouse, we see that there are certain cellular changes that cause glymphatic function. Do we see those changes in human tissue? Are there genetic associations between gene variants related to glymphatic function and Alzheimer's risk. So for example, if aquaporin 4 defines glymphatic function, if glymphatic function contributes to Alzheimer's disease, then you would expect that naturally occurring variation in the aquaporin 4 gene, the human gene, would confer either risk or protection from Alzheimer's disease risk or its different features. Do we see evidence that glymphatic function contributes to the clearance of amyloid and tau from the human brain? Good question. Do we see evidence that glymphatic function is impaired in these types of settings? So in the aging human brain, in the setting of cerebrovascular disease, in traumatic brain injury? And last, do we see that if we modulate glymphatic function in the human brain, do we improve amyloid and tau clearance? Do we reduce the development or slow development of amyloid and tau pathology? So some of these we've got good answers to. Some of them we have less good answers to. So I'm going to zip through a couple of interesting findings on our way. So first, the histological question: do we see changes in the human brain that in mice are related to glymphatic dysfunction? The answer to that seems to be yes. So in mice, we observe that aquaporin 4, which is perivascular in its localization, so this is a single capillary, this is the aquaporin 4 along it, we see that perivascular localization is lost, and we see that in mice, loss of perivascular localization impairs glymphatic function and promotes the development of Alzheimer's pathology. If we look at perivascular localization of aquaporin 4 in human frontal cortical gray matter, that's shown here, we see that there's a certain amount of perivascular localization that we can quantify. We observe in cognitively normal individuals versus individuals who die with Alzheimer's disease, with histopathologically confirmed Alzheimer's disease, we do see that there is a decline in perivascular localization. So that at least is consistent with what we see in rodents. This is one example. There have actually been several papers that have looked at changes in perivascular aquaporin 4 localization across different Alzheimer's disease risk factors or different Alzheimer's disease case series. If that's something you're interested in, you can go check that out at this QR code. So we do see changes in the human brain that correspond with what we see in the rodent brain in terms of histopathological changes associated with glymphatic dysfunction. What about genetic associations? Well, we know that aquaporin 4 maintains its perivascular localization through its anchoring with the dystrophin-associated complex. So that's a set of proteins, including alpha-syntrophin, dystrorevin, dystrophin, and dystroglycan. These maintain its perivascular localization. We know that each of these genes has a number of naturally occurring variants in them called single nucleotide polymorphisms, or SNPs. What we did is we looked into the ADNE1 cohort. We looked at different tag SNPs in the aquaporin 4 gene and the dystrorevin gene. We looked at the SNPs that were available in the ADNE1 cohort in the dystroglycan and the alpha-syntrophin gene. And we evaluated whether these variants were associated with either CSF80 biomarkers, longitudinal cognitive decline, or longitudinal atrophy of the cortex or hippocampus in a multivariate model that corrected for various confounding variables. The results of this study we haven't published this yet. It's in a zillion paper tables that you don't want to look at. This is the punchline. The punchline is that of the 11 tag SNPs in the aquaporin 4 gene that we looked at, 3 of them were associated with variability in CSF tau and phospho-tau levels, or with cortical or hippocampal atrophy. Of the 40 tag SNPs in the dystrorevin gene that we looked at, 15 of them, so fully a third of them, were associated with variability in CSF80 biomarkers, rates of cognitive decline, and rates of cortical and hippocampal atrophy. Of the 4 SNPs in the dystroglycan gene we looked at, 2 of them were associated with rates of hippocampal atrophy, and we were only able to look at 1 alpha-syntrophin SNP. It was associated with variability in the CSF amyloid beta 42 levels. Other groups have reported similar genetic associations, always with the aquaporin 4 gene. So no one else has yet looked at these other genes with regard to Alzheimer's disease risk. But a number of studies now have reported associations with aquaporin 4 SNPs related to Alzheimer's disease and sleep. So you can go check that out here. Do we see evidence that glymphatic function contributes to the clearance of amyloid and tau from the human brain? That's a little bit tricky. To be honest, I would say there's a little bit of data, but it's not very good yet. This is one piece of sort of sideways. I wouldn't call this conclusive. I would call this circumstantial data. So this is a really interesting piece of work from Brendan Lucy, who's at Washington University in St. Louis. So this was a crossover study in only 5 individuals, so 5 participants. What they're doing is serial CSF sampling and serial plasma sampling across the day-night cycle. One of the things they have in participants who either undergo normal sleep or enforced sleep deprivation. So it's a crossover design, so participants are their own controls. One of the things they observed is that with sleep deprivation, CSF levels of biomarkers go up, while plasma levels of biomarkers go down, right? Which is interesting. And if they look at the ratios of CSF to plasma biomarker levels of A-beta 40, 42, total tau, and phosphorylated tau, they observed that what happens with sleep deprivation is that the CSF to plasma ratio is uniformly increased with sleep deprivation. And what they think that this reflects is an impairment of the CSF to plasma clearance of these biomarkers with sleep deprivation, suggesting that the clearance of amyloid and tau from the CSF to the plasma is sensitive to sleep deprivation. Does that prove that that the glymphatic system contributes to the clearance of amyloid and tau? No, but it's at least consistent with it. So we'll give that a yellow check. We'll give that a maybe. Next, is glymphatic function impaired in the setting of Alzheimer's or setting of AD risk factors? This is a tricky question because it comes down to the question of how you measure glymphatic function in people. As I mentioned before, contrast-enhanced MRI, dynamic contrast-enhanced MRI, is the gold standard approach for measuring glymphatic function. But there are many different MRI-based measures using structural MRI to measure MRI visible perivascular spaces, diffusion-based approaches, whether the DTI-ALPS method or multi-shell diffusion-based approaches, perfusion-based approaches that are based on diffusion-prepared ASL, some fMRI-based approaches. Laura Lewis is a prime example of this, what she's doing, that are all believed to be related to different facets of cranial fluid dynamics. The only problem is we don't know what the relationship is between these different measures of cranial fluid dynamics and glymphatic solute exchange measured by contrast-enhanced MRI, right? So many of these methods haven't been validated against the gold standard method. So at this point, while we do see that many of these are altered in the setting of Alzheimer's disease or in the setting of risk factors, we don't really know what their relationship is to glymphatic function. So I would say that the jury's still out on that one. Last, if we modulate glymphatic function, do we change amyloid and tau clearance and pathology? So if I had a drug that if I gave it to people, it increases clearance, does it clear amyloid and tau? Does it improve pathology? And the answer to that is, well, if I had the answer to that, I'd be on a yacht in the Caribbean and I wouldn't be giving this talk right now. So we actually don't have an answer to that. At this point, no one has yet demonstrated that any particular drug in people enhances glymphatic function. And to this point, no one has demonstrated that enhancing glymphatic function in people improves the clearance of amyloid and tau and reduces pathological burden. That's sort of a smoking gun type of question. So we're not there yet. So does glymphatic dysfunction contribute to the development of Alzheimer's disease? We actually don't know yet. To date, at this point, there's strong preclinical data and quite a bit of supporting corroborating clinical data supports a role for glymphatic dysfunction in Alzheimer's disease, but the mechanistic causal data has not yet been demonstrated. Last idea here, why does this matter? This matters because of where we think lymphatic dysfunction sits in the neuropathological Alzheimer's disease progression. So this is something called the Jack curve. What this reflects is what is believed to be the stereotyped progression of the things that eventually lead to cognitive and functional decline in Alzheimer's disease. So that's the last thing that happens over here on the right. Decades before the memory and functional impairment that lands a person in the memory clinic, decades before that, is detectable. You have the development of detectable levels of tau and amyloid pathology. So if glymphatic function contributes to amyloid and tau clearance, and glymphatic impairment contributes to the development of amyloid and tau pathology, then its impairment sits to the left on this Jack curve. And what that means is if we could measure glymphatic impairment, either with MRI, with a device, with a fluid biomarker, then we could identify people not who have amyloid and tau pathology already, but people who are at risk for developing it in the future. And if you could define different therapeutic approaches, whether pharmacological or otherwise, that enhance glymphatic impairment in these vulnerable individuals, then that opens the door not just to secondary prevention of Alzheimer's disease by clearing away pathology, but actually to primary prevention, which is prevention of the development of that pathology in the first place in vulnerable individuals. And that's the big upside that people are thinking about when they're thinking about glymphatic function and Alzheimer's disease. So with that, I'll wrap up, take some questions. This is my lab. These are our collaborators. This is our funding. We love collaborating with smart people who are doing work adjacent to us. So if you're interested in talking more about this, this is my email address. This is my lab's website. This QR code will take you there if you want to chat more in the future. But for now, I'm glad to take questions if you guys have any. Thank you. All right, I see a question from Yuko.
Hi, thank you so much. That was a really interesting talk. Can you talk a little bit about how things like sleep quality, sleep apnea play into this pathway? And if you think that assistive devices like the CPAP and all that would change this function?
That's a great question. So when you think about the relationship between sleep and glymphatic function, there's actually two dimensions to it. One is, if you could sleep, what kind of function would you have, right? And you can imagine impairment of glymphatic function that affected that. That's what we think aquaporin 4 is doing. Like if aquaporin 4 becomes mislocalized, then even if you slept well, we would expect function would be reduced. But there's a second piece, which is, what if you can't sleep well, right? Even though even if all the machinery was intact, if you can't achieve the states of sleep that are where this is being driven, then you could have sort of this secondary effect on glymphatic impairment. And we think that sleep apnea is probably doing both. We suspect that the hypoxia and hypercapnia that you have with sleep apnea is probably damaging the glymphatic system itself at a molecular level, but then also the sleep disruption that comes with it, right? So the fragmentation and the shallowing of sleep that occurs is almost certainly impairing the opportunity for function as well. A paper just came out so there's different MRI approaches for measuring glymphatic function. The use of structural MRI to measure MRI visible perivascular spaces is very widely used. It's unclear how this exactly relates to glymphatic function, but a lot of us are using it. There's a really interesting paper that showed that people who were adherent to CPAP for 6 months, so people who were non-adherent to CPAP, so people with OSA, exhibited progression, so greater and greater MRI visible perivascular spaces, so worse and worse function, if that is a measure of function. Whereas people who were adherent to CPAP, that decline was arrested. So they saw no change in perivascular space burden. That's some of the very earliest data that suggests that CPAP itself may be a way to preserve glymphatic function in humans. We have an active study here using a couple of other modalities where we're looking pre-post, looking at cross-sectionally at OSA itself, and then looking longitudinally with CPAP also. But that's not going to read out for another year or two. But there's some early data that suggests that it's impaired in the setting of OSA, and CPAP seems to help things. There was
I have a question.
Yep.
Dr. Chernishev from Shreveport, Louisiana. Question is kind of in continuation to this. What do you think if we use CPAP as a control group, like in healthy individuals without obstructive sleep apnea? That
Yeah.
might help clearance of the lymphatic system. And second, also related question, because we're all talking about process S, process C2, process model, and adenosine is blamed for development of sleepiness. Do we have any evidence that lymphatic system is doing, clearing adenosine?
Yep. So control participants in sleep apnea. So we had a pilot part of our CPAP study, and we used sleep clinic patients who didn't end up having OSA. We tried to use them as controls. We didn't see big cross-sectional differences between them. The problem is those are people that have other sleep issues, right? So they have insomnia and they have other things. And so thinking carefully about who the appropriate control is, at least in a cross-sectional study of OSA, I think is a reasonable question. I think, quote unquote, healthy people might be a good idea. I think having BMI matched and sex matched people probably will matter a little bit. I think looking for a dose response relationship of an AHI index type of thing, like how bad your OSA is, might be sort of continuously related with evidence of impaired glymphatic function. So that might be a way to deal with that control question, is looking for a dose response relationship. So I think it's a little bit tricky, as all clinical studies are. Adenosine is a really interesting candidate. At this point, there just hasn't been a lot of work trying to understand which molecules are regulating this. There's a very strong case for central norepinephrine in this process, and that's well developed. When I think about what are possible modulators of glymphatic function, the things that I think about are sleep-wake regulating molecules that are also vasoactive and also serve as modulators in neural function. Norepinephrine is obviously one of those. Adenosine is obviously one of those. At this point, no one has yet tested whether adenosine signaling regulates glymphatic function. I think it's a matter of time before someone does. I think that's a very strong candidate. It's just that no one has yet asked the question.
Well, it would be interesting to see for orexin. Orexin, that's one.
Yep.
Because orexin, you can try orexin antagonists,
Yep.
and see what it will do. Interesting thing. You were discussing, very well discussion. I really enjoyed your presentation. Very, very talented, very talented. Well, important thing is, like when you're talking about anesthesia and chemical sleep, have you ever tried to see how the lymphatic system behaves if you do sodium oxybate, for example?
Yeah. So at this point, no one has yet looked at sodium oxybate. I know, I suspect Jazz Pharmaceuticals is interested in that question. Brendan Lucy at Washington University has been doing some work on those sort of that. So they have that serial CSF and plasma sampling regime that they do there. It's very complicated. It's probably very expensive. It sounds horrible, but it's really valuable data. I know that he has been doing some work with DORAs there that seems really interesting. I would be shocked if he has published some work with oxybate, but in that study, it was in JAMA Neurology, I think, in their hands, the drug itself didn't have a big effect on sleep itself. And so it's had a very variable effect on CSF dynamics of amyloid and tau. And so it was a very equivocal finding. I thought it was a kind of a weak finding. And so I think the jury's a little bit still out, but I think that there's definitely interest in oxybate alongside the DORAs, for sure.
What is your take on DTI ALPS index measurement? Because it has been so localized around the medullary vein that, and when we correlate the DTI data with any pathology, people always
question. It's associated with everything. Yeah. So I want to say this respectfully, because many times in an audience like this, there are people who are using the DTI ALPS method. I don't think, and key MRI researchers in the field don't think that DTI ALPS is measuring glymphatic function. And there's several reasons why we think that's true. And there's actually a couple of really important reviews that are going to be coming out here really soon that get at this. So first, the very ubiquity of associations is problematic, right? The fact that DTI ALPS changes in that index are associated with literally everything suggests that it may not be measuring anything, right? Or it's measuring something that's so ubiquitous that it's probably not related to clearance. Second, we think that the B values on MR that are sensitized, that are used for DTI, so typically around 1,000, are probably not sensitized to the slow extracellular flow that we're expecting to see. So we think that they're probably sensitized to changes in the axonal and not the perivascular compartment. Third, if you actually look at the data from all the DTI ALPS study, and one of the studies that's going to come out is it's a meta-analysis. So the DTI ALPS is an index. So it has the axonal compartment divided over the vascular compartment. The change in the vast, vast, vast majority of those studies is in the axonal compartment, not in the vascular compartment. It's just that you don't see that in the index. So people are just reporting the index value. And so the fact that it's all in the axonal compartment tells you what this is picking up is subtle changes in axonal white matter architecture and probably not perivascular exchange. Lastly, that Oslo group that's been measuring glymphatic function with contrast enhanced MRI, they've now done it in hundreds of people. And they've also measured DTI ALPS in those hundreds of people. And my understanding is that there is a paper coming out soon that shows that there's no correlation between the two, right? So for that reason, I do not think that the DTI ALPS measure is measuring glymphatic function. It is measuring something about white matter structure that is and it's doing so in a very sensitive manner, but it's not glymphatic function. So sorry to anyone who's doing that in your at your institution.
So if you have like the patient who have autoregulation, do you think like the glymphatic system will change?
Yeah, that's actually a super question. So we see in animals that changing blood pressure acutely does change it. We see that evoked vasodilation does impair it by closing off perivascular spaces. To the extent that autoregulation reflects sort of the integrated relationship between changes in blood pressure and cerebral blood volume and flow, I suspect that in patients with altered autoregulation, you would expect to see altered exchange. No one has done that study.
Thank you.
Omar Hassan.
Oh, sorry. I actually had the exact same question about ALPS. We both work in similar groups. I guess I do have another, and I apologize if this was addressed already, question about whether glymphatic function or diffusivity, I guess, changes depending on, or if there are any differences between regions of the brain?
Yeah. So glymphatic function, I would say it is not synonymous with diffusion. So I would say glymphatic function involves its exchange between the CSF and the interstitial compartment. It involves a diffusive component, as all transport does, but it also involves, at times and in places, a convective component also, so both. There is good evidence to suggest that it is, and this is really, there is evidence to suggest regionality among gray matter regions. There's also the contrast-based data. One of the findings that people have is that if you inject contrast into animals and into humans, you don't see enhancement of the white matter at all. And that's a known thing. Everyone observes that. And the story on that has always been, oh, there must be less glymphatic function in white matter because we don't see contrast there. But actually, some of us, when we talk about it informally, and this isn't something that you'll find in the papers, this is just sort of in conversation now, we actually think that transport in white matter may be much faster than in the gray matter, and that the absence of enhancement that you see in humans and in animals may actually reflect the fact that as contrast comes in, it's dissipated so quickly by very fast flow that it just doesn't accumulate to levels that you can see it. So one possibility is that white matter is an area of very high transport rates, right? Much higher than in gray matter, which may have something to do with both with its structure, right? But also may have something to do with why, MRI visible paravascular spaces show up in the white matter and not in the gray matter, right? So there may be something very, very different about water or fluid transport in white matter and gray matter. So we do think that there are regional differences, though.
Great, thank you.
Okay. Other questions? We're a little over time. Thanks for good questions and some attention. I think that's what we got. Again, I'm going to just put this up one last time, just as a shameless plug and shill. If you're interested in collaborating, we are not a stroke group, and we are not while my background is in cerebrovascular physiology, I'm not really connected with that anymore. So I think there's some pretty big lanes open for people who want to study glymphatic function in the setting of cerebrovascular disease and stroke. So if you're interested in talking about that, I'd love to talk about it. So thanks, everyone.
Yeah,
Thank
this
you.
report we're very interested here. Yes, thank you.
All right, take care.
Thank you.